Kia ora everybody. So I'm not actually a doctorate student, I'm still doing my master's. My background is in biomedical science with a focus on microbial pathology and genetics. So apologies if it's quite technical. And so my project is about enhancing roof water safety using metagenomics and qPCR in the Ngāi Tahu Takiwā. So just a bit of background. So my project came about due to a community concern about the impact of birds on roof collected drinking water. So it takes place in a coastal community that is not on reticulated supply, and they rely on other methods to collect their drinking water. So they haven't had it the easiest when it comes to drinking water, as they've got high rates of nitrates in groundwater sources. So because of this, some residents are using roof water as a viable option for collecting drinking water. But many expressed concerns about the impacts of bird life on the quality of the drinking water. So the surrounding area already boasts a really large bird population, and due to changes in the harbour's ecology, they have had an increased frequency of mass feeding events, and during these mass feeding events, the population of bird life increases, with residents remarking around 10,000 more birds, and then the birds land on the roofs, leave droppings, and then the droppings get into the roof water supplies. So many in the community raised concerns about contamination and the risk of illness due to this, and expressed the desire for more information about the safety of the drinking water. So the aims of my project is to help address this community identified need, to contribute data to support safer water use in roof collected supplies. The findings aim to inform water safety planning and empower local runanga with evidence to guide risk management decisions. So this community highlights ongoing drinking water inequalities as they rely on their own private supply and have limited council support. So it's a clear example of the need for targeted, locally informed water safety interventions. So by applying metagenomic sequencing and qPCR to detect a broad range of bacteria, we hope to uncover various patterns and risks that aren't always apparent with traditional water safety testing measures. And we also aim to compare bacterial community profiles before and after household level treatment to see how effectively they reduce bacterial loads and potential health risks. And the hope for this is to just provide a bit more information about the treatment options that are available for community members and the effectiveness of these options. So my supervisor, Tim Chambers, and we went to collect water samples alongside a runanga representative who had been trained in water sampling. So we collected samples from 15 different roof water supplies. These sites all differed in treatment type, maintenance history, and infrastructure. Literally not a single site was the same. Many sites had no treatment at all. Some of those that did used only filters, whereas others had a much more comprehensive system that included UV and filters. So at each site we collected 100 ml samples for the routine water testing and 1,000 ml samples for metagenomics. Sites with no treatment were sampled just at the tap where they collect most of their drinking water, and sites with treatment were sampled at the tank before treatment, and at the tap they collect most of their drinking water for after treatment. So first we did the standard water testing, which is testing for E. coli and total coliform counts. So this revealed widespread contamination within all of our sites. All the values were highly variable, and some sites much more contaminated than others. So for the untreated samples, which are these ones here, 100% of our sites tested above the acceptable value for total coliforms, which indicate environmental contamination, and 60% tested above the acceptable value for E. coli, which indicates fecal contamination. We also had 100% of our filtration sites detect above for both E. coli and total coliforms. However, we only had 2 of these sites, and there are a lot of factors that can influence this. And then for our sites with UV and filtration, None of them tested above for the acceptable value for E. coli, and only one tested positive for any total coliforms. And so after this, we underwent metagenomic analysis. So metagenomics is the study of genetic material collected directly from environmental samples without having to grow the organisms in a lab first. So traditional microbiology often requires culturing microbes, but many won't grow in a lab, or it's really hard to. So metagenomics can get around that by extracting all the DNA in a sample at once, sequencing it, and then using computational tools to figure out what organisms are present. So this approach lets us detect the full diversity of microbial communities, including rare or previously unknown species. In short, it's like taking a genetic snapshot of the entire microbial ecosystem. And for my study, we focused on just the bacterial community and targeted the conserved 16S rRNA region that is found in all bacteria. So we had two types of DNA extraction methods. So the first, which is the filter pipeline, that is just directly sequencing all the DNA found in the filtered water. So that it just gives us all the bacteria that are present at the time of sampling. And then the second one was we ran colilert tests on that same water, and if they were positive for E. coli, we then took the growth media, grew everything on there that was in the water, and then sampled the growth media and sequenced that DNA. So that enriches for any samples that grow in the same conditions as E. coli. And so that gives us just a bit more of a targeted look at any bacteria that are linked to potential fecal contamination. So by combining the two approaches, we can see that both the overall bacterial diversity in water and the specific organisms enriched during contamination events. So this figure here is just showing some of our filtered samples. So it's basically the bacterial fingerprint of roof water. Each bar is a different sample, and the colours represent the different bacteria living there. So you can see that each sample is made up of many different genera, but the mix changes depending on the site. So each roof essentially has its own unique bacterial community. The grey bars here are all the other bacteria that didn't make the top 19 in abundance, so that's just everything else as well. So the large proportion of other shows that the roof samples are highly diverse and not dominated by just a few well-known genera. And this just demonstrates the richness and complexity of the bacterial community. And then what's also important is that we detected Legionella in these samples. So this is a well-known pathogen that causes serious respiratory illness. And so finding it in this roof water tells us that just standard E. coli testing is not going to reveal all the health risks that are associated with roof water. So basically it just tells us that roof water is a very diverse bacterial community, and it does include potential pathogens and highlights risks standard monitoring alone would miss. So then this one here is our colilert sample. So when we selected for bacteria that grow under the same conditions as E. coli. What's important is that we don't just see E. coli, but we see a wide range of bacteria that also thrive in these conditions. Among them, some well-known pathogens, Salmonella and Shigella there, which are associated with serious gastrointestinal disease. And as well as this, almost all of the other bacteria that appear in the top abundance here are opportunistic pathogens. So given the chance, they can cause illness, and in particular with the most vulnerable members of our community, such as infants, elderly, and the immunocompromised. And so what really this figure is telling us is that when we enrich for E. coli, we're also enriching for a whole group of potentially harmful bacteria that are also found in the roof samples. And again, relying on E. coli alone as an indicator may underestimate the broader health risks. And while genomic approaches reveal the wider community of pathogens and opportunists that also exist in roof water. And then so after that, we also use qPCR for fecal source tracking. So then this adds just another layer of understanding. While metagenomics tells us what's in it, qPCR can be used to target specific bacteria. And so we've used it to identify what animals are contributing to the bacterial load through their feces. So DNA is extracted, and then qPCR targets bind bacterial markers that are unique to certain animals. So like there's a specific bacteria only found in humans or birds, livestock. And then we can link the presence of bacteria to specific contamination sources. So we tested 13 of our samples with qPCR, and of 8 of them were positive for the GFD marker, which indicates that bird contamination. And we also had, which is supportive of the community's concern about the birds influencing its water quality, and none of our samples tested positive for any of the other markers related to humans or ruminants or dogs. I mean, you'd be a bit surprised if some of them did, but we tested for them anyway. And then we also targeted the general fecal marker, which is GenBac, which just tells us if there was recent fecal contamination. And most of our sites tested had this detected. And this meant that fecal bacteria was pretty widespread within our sites. And it also just highlights again where E. coli falls short, as some of them tested below the acceptable value for E. coli, but GenBac was still detected in these samples. So with just E. coli, it says no, there's not fecal contamination, but using GenBac, it said yes, there is fecal contamination. And then lastly, after this, we compared bacterial reductions across 3 different treatment sites to see the differences before and after treatment. So the UV and filters was the most effective treatment that anyone in the community had, and both sites here were tested below the acceptable value for E. coli after treatment. However, there is differences in the efficiency. This site here, site 5, the resident was very diligent with maintenance. She made sure she was always replacing the filters every 6 months and the bulb every year, because she was right on the coast, so she was quite aware of those risks. And you can see that there was a 93.2% decrease in the total bacterial reads between the 2 samples. And then we also had site 10, which was also UV and filters, but that one only decreased by, only 60%, just sort of showing some differences in efficiency between systems. And then lastly, site 14 had only filters, and there was only a decrease of 17.5%. And that just kind of highlights that partial treatment is not as effective as a full comprehensive system. And so this just highlighted that filtration plus UV plus regular maintenance is key to effectively reducing the bacterial contamination in roof water. And so the next steps for me is just conducting another round of water testing, and this one is going to be after a period of low bird activity. And then this is going to allow us to compare for a comparative analysis of bacterial diversity and the abundance of pathogenic bacteria between high and low bird activity periods. And by comparing these 2 time points, we aim to better understand the extent to which bird presence influences bacterial contamination that can potentially lead to illness. And ultimately, want to generate locally relevant evidence that supports safer water use, empowers runanga decision making, and addresses ongoing drinking water challenges faced by communities on private water supply.